Really excited to welcome Professor John Paul, who has the dubious honour of having been my longest collaborator. For over 15 years we've worked together on microbiology projects. But also he has a long and illustrious career in microbiology as well, which is very... you're very humble about usually, so I will tell you. He's the professor, honorary professor at Brighton and Sussex Medical School. He formerly was the lead microbiologist for Public Health England for the southeast of England, which means he was in charge of all the microbiology, infectious diseases stuff for the south of England. And also you've worked internationally, having done projects in places like Nairobi in your earlier career. Very interesting stories. And as a part-time, soon to be more full-time, bacteria tourist, which he'll be explaining to us, and possibly trying to encourage us to become bacteria tourists ourselves, I think. So without further ado, I'll hand over to Professor John Paul. Thank you.
Great. So Anna had suggested that I could talk to you for a bit about bacteria in the context of life in the future and in a world without us. So this is going to be a happy talk because as far as bacteria are concerned, conditions are always good for them. Biologically, they're diverse. They've been around for a long time, so the genetic variation amongst all the different groups of bacteria is enormous. Some of the bacteria are able to tolerate extreme conditions, so some of them can tolerate extreme desiccation, they can tolerate ionizing radiation, and not only can some of them tolerate extremes of temperature, but there are groups of bacteria that are thermophiles, so they will grow optimally, for example, in boiling water, and will become dormant, in effect, if they're in what we would consider to be ambient conditions. And across the wide range of bacteria, they can exploit all manner of food sources in ways that we find almost unimaginable. They can form spores and become dormant for very long periods. And even if they don't form spores, under certain circumstances, they can adapt to very low replication and turnover rates. So, for example, as bacteria fall onto the seafloor and become compressed into sediments, and those sediments gradually start turning into rocks, what you end up with is populations of highly selected bacteria that have become constituents of the rocks, in effect, and no one really knows what their replication time is, but it must be very long. And they're not quite dead. They're not really doing very much. They're sort of undead bacteria. So basically, to sum that up, there are always going to be opportunities for them, even in conditions that we would consider to be way too extreme to sustain human or mammalian life. Right. So Anna's come up with some sort of what sounds like a sort of fictional account of me, and the real story is that I trained as a medical microbiologist, so my career, in effect, has been around working in hospital or in public health labs and paying attention to a very narrow set of organisms that cause infections in humans. And there aren't very many of those. Compared to the huge extent of biological diversity that there is in bacteria. And I mean, that's great, because it's a fun career to do, but one of the frustrations of that is that in my spare time I like to take an interest in natural history. So I'm a keen entomologist and I like to be out in the field. And as I progressed through my training and through my career, I started to think, well, and as I developed an affinity for dealing with bacteria, I started to feel frustrated that I didn't understand what their role was in the natural environment. So I became a self-styled bacteria tourist. And I've taken some pains over the last number of years to go to places in the world where you can see interesting bacteria. So the top slide is represents what I've been doing for a living for my career, which is a slide from a medical sample showing pneumococci, Streptococcus pneumoniae, from a medical sample. And that's the bug that tends to be a common cause of pneumonia. And the sort of test you might do would be to grow the bug and do a gram stain and identify it and do antibiotic susceptibilities. And that's interesting. That takes you into its own little world in itself. And then as a bacteria tourist, it's fun to go to places like Yellowstone Park and go to the hot springs there that are at boiling point and study the literature about the bacteria that thrive in those conditions. Okay, so for those of you who are not microbiologists and would like to have some sort of grasp of the world of bacteria, we'll just go through this slide. The word is from a Greek term for a little stick, and bacteria are very small. So a typical bacterium, such as the one Escherichia coli, which is part of our gut flora, is much less than a millimeter in length. It's microscopic. There are lots of bacterial cells in the world. If you look at Karl Mayer's paper, you can see how that estimate has been reached, but it's a very big number, an unimaginably big number. A gram of soil may contain 10 billion microbes, and they're pretty much everywhere. So, surprisingly, you might think of when you think of the rest of life, like plants or animals, we tend to think of places like rainforests as being unusually diverse and rich. And it's actually been found from the evidence that's currently available that deserts are among the richest places for bacterial diversity. Hot springs have their own particular floras. Bacteria can exist on and in ice. They occur in deep sediments, and as I've said, even deep boreholes, rock samples, yield bacteria that are living deep down in the earth. There are bacteria that live in high salt concentrations, and even oil wells, when oil is extracted that's been underground for all those millions of years, that sometimes has bacterial floras. And there's some question there about really how the bacteria that live in petroleum reservoirs get there. Some of them, no doubt, get in as the wells are opened up, but some of them have probably been there all that time. And it almost invites quite sort of controversial questions like maybe we should consider petroleum reservoirs to be nature reserves and to preserve them for their unique microbial floras, as we have nature reserves that define other kinds of habitat. A significant proportion of the Earth's biomass is made up of the organic carbon that's in bacteria. Now, one question that we haven't got an answer for yet is how many species there are. So certain kinds of habitat have been looked at very exhaustively and are beginning to plateau off. So if you imagine how we start to develop estimates for, I don't know, birds in a particular habitat, you do something called a collector's curve and spend a lot of time monitoring the birds in a particular place and make a list, and you can plot a graph and you can see as it starts to plateau, you can reach some confidence that maybe you're getting to the point where you've got an idea of what's there is what's there. With bacteria, maybe we're getting close to that with the human mouth flora as being about 700 species. But for places like the soil, or if you look at an Amazonian forest soil sample or whatever, and keep going back and looking at it, when you look at the literature, you get an idea that it's still the collector's curve is still a straight line, so we can't really estimate how many species there are in a particular rich habitat, let alone what the global population of species is. Anyway, it doesn't matter how many there are, we just know there are lots. And wherever they are, they constitute a normal flora, which is part of the biology of the planet. only a tiny number of which have become specialized in a way that allows them to cause disease in man. And those, of course, are the ones that we've been particularly focused on for very obvious reasons for the last couple of hundred years, I suppose. So, thinking about where bacteria might be in the future, as speakers have said at this meeting over the past couple of days, if you look back at the past, it might help you understand the future. Now, I suppose a cynical way of looking at that would be to say that what you learn from history is that you don't learn anything from history. But let's assume that you can learn something from it. And we know that bacteria have been around through most of Earth's history. So they've seen it all. They've seen environmental extremes, and they've flourished and prospered through that. And they can exist and flourish in conditions that are hard for other organisms. So they've got very wide range of tolerance to what's on offer. And they can occupy the places that are not fit for other life forms. So that picture there is of the famous stromatolites that occur at Shark Bay in Australia. And that's a place where there's a saline lagoon that hasn't got that many advanced life forms, as you would say. And in those undisturbed waters, films of bacteria grow and encourage precipitation of minerals and carbonation of the film that they're in. And then the bacteria then migrate up through the film as it grows. And gradually what you get are living rocks, which are stromatolites. And this is one of the few places in the world where you can see stromatolites today. But in previous parts of Earth history where there weren't other big animals to disturb microbial films, there are evidence of big stromatolite beds, and that gives you an idea that there are periods when bacteria have sort of dominated the living world. Now, if we do disappear in the future, and some people predict that humans will disappear at some time in the future, then that's not good for the ones that are adapted to live on us, because part of our normal flora, we have a normal flora of bacteria. As I've said, there are maybe 700 species of bacteria that live in the mouth. There are doubtless many more that live in the gut, for example. They're part of us. Mammals have co-evolved with their microbial flora, and some people are starting to think of us and our microbial inhabitants as a sort of consortium that are living together in a kind of harmony. So it's not good for them. So the estimates based on looking at very old zircons, little minerals in the Earth's oldest rocks, I think from Greenland and places, that are thought to have been formed around the time of the formation of the solar system and the accretion of various bits of dust to form the planet, and looking at the decay of uranium isotopes in those zircons, have estimated that the solar system and Earth were formed roughly 4.5 billion years ago. And then there's evidence only a few 100 million years after that event, as things have started to cool down, based on the composition of rocks, I think, in Greenland that have got traces of an organic presence to them, that maybe life has been around for about 4 billion years. Now, how it got to Earth, how it arose, of course, is one of these things that open to speculation. And either it evolved in some warm pond, as Darwin said, in situ, or there are people who think that there's a strong case for panspermia, that life has come from somewhere else. Be that as it may, it's been around for a long time. And by 3.5 billion years ago, there are fossil stromatolites in Western Australia that look like current-day living stromatolites, and also some authors have put a fairly robust case together looking at electron micrographs of those rock samples, that there are filaments of the remains of fossilized bacteria in them as well. So the consensus is that they've been around for at least 3.5 billion years, and maybe 4. And then various things have happened through Earth history that have been really quite planet-changing. So there's evidence that about 2 billion years ago there was a period when there was a big change on Earth, and that's encoded in what are called the banded iron deposits, where there are massive layers of iron ore that have been deposited with, which occur in bands. And that's just a property of the way that these layers have been put down. And that's thought to reflect the fact that the planet was becoming oxygenated because bacteria had become photosynthetic. Bacteria had worked out how to use the sun's energy as an energy source to fix atmospheric carbon dioxide into organic carbon to build their body parts. And that because they were using water as a reducing agent and liberating oxygen, it was gradually oxygenating the atmosphere. Well, first of all, it oxygenated the sea and caused ferrous iron to precipitate out as these big rust deposits in the sea. And then it went on to oxygenate the Earth. And that's widely hailed as being a great thing for the evolution of life on Earth because it led to aerobic organisms like ourselves. But it must have been a very bad time for the anaerobic bacteria that are around then, because the anaerobic bacteria, some of them would have found oxygen a very reactive, dangerous molecule to be in association with, and the anaerobes have had to retreat into the anoxic recesses, like into sediments and into our gut flora and places like that. So good time for us, but bad time for anaerobes. And then there have been various times when so much when so much carbon dioxide has been locked up, it seems that there's been massive global cooling, resulting in what has been popularly described as Snowball Earth situations, where the Earth has drastically cooled and there's been glaciation down to the tropics. And that provides opportunities for bacteria as well. It provides opportunities for psychrophiles, and in the aftermath of those Snowball Earth events, in the massive change that have ensued, that's also been a spur to new forms of life. And one of those, the last Snowball Earth, occurred sometime before the Cambrian radiation. And that was the last really big biological event, I suppose, in that when everything sort of melted and normalized before the Cambrian period, there seems to have been so much opportunities available in terms of food and ecological space, as it were, for a massive appearance of complex animals for the first time that eventually led to vertebrates and then mammals and us. There have been several mass extinctions through history. And one of the interesting things about mass extinctions when it involves big animals is that although we think that's rather a tragic event, so there was a big mass extinction at the end of the Permian, and of course we know about the dinosaurs, but when you get these trouble spots in history, that's when you see little periods of big stromatolite beds. So those are obviously good times for bacteria because they're able to go about their business undisturbed and form biofilms that form into sort of big rock-like structures, and then we can still see them in rocks today as evidence of a big boom time for bacteria. And of course, we're fairly recent in all of this, but we're sentient beings, and we have the luxury of being able to digest all of this information and wonder what it's all about. These are some of Anna's paintings that I like to use. So these are the main bits of life as far as we are concerned. There are the little creatures, the prokaryotes, the ones that don't have a nucleus, that are basically little sort of bags of DNA and a few little organelles in there, which include the bacteria, and another group called the archaea, which many scientists consider to be a separate branch of life, although they look much the same. And then there are these complex big cells, the eukaryotic cells, some of which are single-celled organisms, some of which get together and they gang up together and they become things like mammals or us or whatever. And if you look at the molecular basis of these organisms, you can see that there are three different groups. But eukaryotes are big, complicated things. Now, there isn't a clear consensus on how we look at the classification of life. The dominant paradigm for the last few decades has been the three-domain model, which was proposed by an American biologist called Carl Woese. And he had looked at ribosomes, or he looked at ribosomal RNA, which is one of the essential nucleic acids involved in protein transcription. And he realized that by looking at RNA, the organisms tended to fall into one of three very discrete patterns. And he said, well, that's a marker that there are three branches of life that must have branched off way deep in prehistory. And one of them was clearly the bacteria. Another one is all the eukaryotes, which includes us and mammals and reptiles and amoebae and other big-celled animals. And then, surprisingly at the time, he said the archaea, which at that time were thought to be a little sort of curiosity of some, were thought to be some extreme bacteria at the time. He said, well, they're a different group, and they've split off early. And there's a counterclaim, mainly advanced by another American biologist called James Lake, who said, well, it's more complicated than that. Organisms in history have been able to swap DNA between different lineages, and it's a bit mixed up. And he put forward some evidence to say, well, actually, the archaea are a group that branched off rather later, and that of the living archaea, the one that most resembles the one that branched off and gave rise to the eukaryotes was a thing called Sulphurs, which is a little organism that lives in acidic, hot sort of swamps, such as in the Yellowstone Park. What's rather nice when you go to Yellowstone as a bacteria tourist is that when you go to this, there's one particular site called Roaring Mountain, and they even have little placards up to tell you what kind of microorganisms are there. So I know everybody goes there to see the grizzly bears and the bison and everything, and it's hard to resist. It's hard to resist seeing the megafauna, but it's almost more exciting to see the microbes. So, what I've just alluded to is that there's the concept of what you might call a universal common ancestor, and if we as life forms, including the bacteria and everything else, are all related, then we'll trace our ancestry back to maybe Darwin's little pond, and say, well, there was some kind of organism or community of organisms at some time that gave rise to these three branches. There was a paper in Nature Microbiology a couple of years ago where a group had looked at, they'd done a trawl through all the sort of genes in the record to try and see what things had in common, and that they suppose that there are maybe about 350 genes that we all have in common, and use that to have a sort of best guess at what that common ancestor was like. And obviously it was a bacteria-like single-celled thing that maybe lived about 4,000 years ago. Now there's a curious record here called From Bacteria. I do find it rather fun to look at how bacteria find their way into popular culture. And there is, if you do a trawl of the internet, you will find there's quite a lot of recordings that have used bacteria as an inspiration. This one has the title track and the lyrics on the actual album sleeve. And as you can see, it says that when bacteria dominated the earth and there were new humans. Anyway, so I'm going to take you through some of the ancient bacteria that you can go and see now and experience. Because Australia is an ancient landmass, it tends to preserve some of the oldest rocks on Earth in reasonably intact condition. They haven't been horribly sort of cooked or damaged in any way. And the Museum of Western Australia has got beautiful stromatolite displays that have been gathered from locally. And on display there, you can go and see a piece of the 3.5 billion year old stromatolites that at the moment more or less has the record for being the oldest evidence of or reasonably uncontrovertible evidence for life on Earth. And then you can compare and contrast that with structures that are being made today. So in what you might consider to be a fairly disturbed environment, like a wet cliff face on an island off the south of England, you can still see evidence that bacteria are there, and they're causing precipitation of calcite. And the picture on the bottom left is a cyanobacterial film, and you can see how the cyanobacteria are growing out as a little sort of mini dome, a little microscopic dome, and they're causing precipitation of calcite. So it's almost like a little mini stromatolite. And once you get your eye in, you can start to appreciate that the bacteria are everywhere, and they're causing some of these structures and appearances that you might otherwise dismiss as just being nothing. But these are caused by living organisms. And then if you go somewhere like the Yellowstone National Park to one of the hot springs, where there aren't any other organisms to disturb the bacteria, you can see little mini stromatolites growing. And then you can travel around the world, and in various places where there are old rocks, you can see actual fossil stromatolites. So on the island of Islay in Scotland, which is famous as a place for production of malt whisky, it's also an excellent place for looking at the geology of the last episode of Snowball Earth, because there's geological evidence there of glaciation occurring in the late Precambrian. And at the end of that period, there are some big stromatolite beds. And what you can see here in these rocks, in the kind of layers, it's a fossilized account of those layers of the bacteria that were being able to grow up unimpaired. And you can also go and see, in Australia, coincidentally, you can go and see examples of living stromatolites. So apart from Shark Bay, there's a place called Lake Thetis, which again is a saline lagoon, which doesn't really have much living in it apart from bacteria. And there you see a couple of bacteria tourists who've gone there to appreciate these life forms. And they've put up a lovely interpretive sign and everything, and it's not been vandalized, and it's a wonderful place to go. And you need to rush there before everybody else gets there when bacteria tourism really takes off. And that's why it hasn't really taken off, is because that's what you get when you get there. But I've saved you the trouble. Now, there's some debate about when life got onto land, but a geologist at St Andrews, Tony Prave, is one of the scientists who has advanced the ideas that it was probably quite a long time ago, because off the west coast of Scotland there are some shoreline, very well preserved ancient shoreline sediments that are preserved in the rocks, and he's found evidence that some of those have microbial mats on them. He's also got electron micrographs showing structures of microbes in these mats, so they do appear to be real. And it's a remarkable place. You can go there, and at a distance it just looks like what you might call a normal bit of rocky coastline. But as you look closely, you can see it is an ancient, beautifully preserved shoreline. You can see the ripple marks that are still there after 1 billion years. And you can see evidence of microbial mats. And even something that you take for granted, like a ripple mark, doesn't form very easily in sterile mud or sterile sand. It's only when there's an organic content and things actually living in it that the things bind together and can be coerced into forming ripples by the wind or the environment. So that's strong evidence that was might not have been anything else there at the time, but that was a microbial, bacterial shoreline 1 billion years ago. And then, whilst you're on the west coast, you can go up and look for evidence of the great oxidation event, when the cyanobacteria started photosynthesizing and oxygenating the sea and causing massive banded iron deposits to form. So the really huge banded iron deposits, again, they're where the really ancient rocks are. They're in Australia, they're in Canada and North America, and you know where all the big iron mines are. But even on the west coast of Scotland, there are some 2 billion year old banded iron deposits that you can go and look at. They're surprisingly heavy things to hold because they're basically iron. And it's thought that the bands are in them because of some kind of cyclical effect on the metabolism of the cyanobacteria, maybe seasonal or even daily cycles when you look at these rocks in close-up. I mean, it looks like a completely ordinary place, but it's got these rocks when you look at it with the right kind of eyes. And then on Islay, where the ferry gets in, there's a cliff face behind the ferry port, which has what are called drop stones in the cliff face. And that's important because drop stones occur when there is massive glaciation. Basically, glaciers scrape off rocks, they transport them, and then when they get out into open water, they melt or the rocks drop out of them and they fall down into silt, and you get this strange geology of what looks like rock that has been formed from sediment, but it's got these inclusions in it that are a different kind of rock. Now, although Scotland is a long way north these days, at the time that these drop stones formed, the people who look at the drifting of continents are able to show that it was actually tropical water at that time, and that's evidence that that was the last Snowball Earth event. And it was after that, when everything melted, that we got the Islay stromatolites forming. Okay. So, bacteria in natural history. That's enough about bacteria in the past. Bacteria in natural history, what do they actually do? So, one of the important things that they do is that they can use light to fix carbon dioxide and form organic carbon compounds to build cells. Now, of course, plants do that, but plants are only able to do that because they have chloroplasts, which are organelles that were originally cyanobacteria that have been included in plants and have gone off to form their own lineage. And then there are many groups of bacteria besides the cyanobacteria that can do photosynthesis. They can also form organic carbon by other means, so by using chemical reactions as a source of energy to fix atmospheric carbon dioxide. So at iron springs, where iron is coming up to the surface dissolved in water, there are bacteria that are able to fix carbon dioxide by using the energy of harnessing the oxidation of iron. And then there are all the other life forms, us included, and many bacteria that have to rely on the primary producers to get our organic carbon. So we're basically stuck with having to eat other things. Then what else can bacteria do? So they can fix atmospheric nitrogen. And nitrogen is a part of amino acids, which is a part of proteins, which is a part of living things. So we're dependent on nitrogen-fixing bacteria. I've already mentioned iron bacteria. And then sulfur is another key element when we're looking at bacteria, because a lot of bacteria are able to harness various reactions related to sulfur chemistry. And then, as I've mentioned earlier, it's very interesting to look at the anaerobic microbes that are not dependent on oxygen, that use other metabolic pathways. And one of the groups of anaerobes are the Methanogens, and they live in anoxic environments, and they're able to get energy by using carbon dioxide, which is released by fermentative organisms, and also hydrogen as a source to form methane. So that might not be a good thing in the context of this symposium, because methane is a more potent global warming gas than carbon dioxide itself. But it's interesting to know that there are archaea, actually, that are able to do that as methanogens. So these are some bacteria for you to look at. These are cyanobacteria. If you go and look at a bluish-green stain on a wall where water's been dripping down, it may contain true algae, but you may be lucky and find it's got some beautiful cyanobacteria in it. Another place where you get cyanobacteria is where there are lots of organic nutrients in water when you get cyanobacterial or so-called blue algal blooms. And that's a picture taken flying over Lake Victoria. And you can see that because of the agricultural runoff and pollution in the lake, there's a massive cyanobacterial bloom occurring. Another visible cyanobacterial form is a thing called nostoc, which is something that's got into folklore. This is an example of the sort of blob and slime that you see suddenly appearing after rain. There are various names in languages. I wouldn't know what it's called in Dutch, but there are terms in English where it's called star jelly or star slime or whatever. I believe the French have called it merde de coucou, and I think in German it's Schleimling. I don't know if there's a Dutch term. I haven't found one in the dictionary. But that's a consortium of bacteria, and when you do microscopy you can see all the little cells of the nostoc bacteria in there. Nostoc is interesting also as a symbiont in the roots of cycads, which you may have as an ornamental plant. And the reason why the cycads have them in the roots is not because they need a photosynthetic partner to make carbon, but they need something to help them fix nitrogen. And nostoc fixes nitrogen as well as a photosynthetic organism. So next time you repot your cycad, have a look to see if you can see these little green rings around the root nodules, which are the symbiotic bacteria. Another plant which has symbiotic cyanobacteria is the water fern, which you sometimes see on pond surfaces. And if you look at the microscopy of water fern, you can see quite clearly that there are the actual fern cells which have chloroplasts in them, which are a bit greener, and then these blue-green ones are the symbiotic cyanobacteria. Another plant is on the left, alder and bog myrtle, both of which grow in boggy conditions where there's poor access to nitrogen. They both have root nodules that accommodate a symbiotic bacteria called Frankia, and that's a nitrogen fixer. And if you pull up an alder tree, you can see it's got these little nodules on the roots which house the Frankia bacteria. And that gives alder the opportunity to be a pioneering plant. So when a glacier retreats, or at the end of the ice age, or when you have a post-industrial site where you've cleaned off all the debris, one of the first trees that will start growing there is alder, and it's because it can grow there in the absence, in very poor nitrogen levels, because it has those symbionts. And, for example, on this very sandy glacial moraine in Canada, you can see that what's growing there is actually alder. Now, I'm sure many of you will have been out for a walk in the countryside or whatever, and been walking through a boggy area and seen these sort of orange slimes on the ground and not really thought very much of it, maybe thought it wasn't very nice, and step over it. But this is a really interesting situation, because this is where iron bacteria are living, and they're exploiting the fact that in the acidic water that's bubbling up there is ferrous iron, and they're catalyzing the reaction to form ferric iron and siphoning off that energy and using that to fix carbon dioxide and build their own body parts. And if you look at the microscope, under a microscope, some of that slime, you suddenly realize this is full of sheaths of Leptothrix iron bacteria. And some sites where iron bacteria are present can be extraordinarily beautiful when you start to shift your aesthetics to appreciate them as such. Another example of having to shift one's aesthetic sense would be coastal sites that stink of sulfur dioxide because of all the decomposing matter. But these are very rich sites for sulfur bacteria. And one of the most beautiful of the sulfur bacteria are the purple sulphurs. And again, these are photosynthetic bacteria. They're using sunlight to fix carbon dioxide, and they have a purple pigment rather than a green pigment to do that. And instead of using water as a reducing agent and releasing oxygen as a byproduct, they're using sulfur dioxide as a reducing agent, and as a byproduct, they're forming elemental sulfur. So when you put one of these under the microscope, you can see the cells of the purple sulfur bacteria, and those little inclusions in the cells are little grains of elemental sulfur that are being formed. And if you want to enjoy sulfur bacteria in your own home, then you can build a Vinogradsky column. So Vinogradsky was a Russian environmental microbiologist, and he's considered to be the first environmental microbiologist. And he was working at about the same time as the great medical microbiologists. But he was more interested in communities of bacteria and how they were using their environments and what the chemical pathways were. And in order to study those pathways, he set up simple columns that involved getting some water and some sediment and providing a carbon source. And the bacteria do it themselves, and you suddenly get these beautiful blooms of sulfur bacteria. And up at the top, then you can get true algae and cyanobacteria as well. And also white sulfur bacteria will form there, which are following a slightly different pathway. I mentioned methanogens earlier. So the places where methanogens are doing very well are anoxic environments where there is fermentation of organic matter that's producing the hydrogen and the carbon dioxide they need. So a peat bog would be the best place to go if you want to experience being in a methanogenic environment. But we humans managed to create pretty much the same environment by dumping rubbish in landfill sites, because basically what we're doing is burying tons of organic matter where fermentation will occur, and we'll set up the ideal place for methanogens to live. And of course the methanogens will then release methane, and we can use the methane as a source of energy. All right, moving on to another environment. Bacteria will flourish in deserts. As long as there's a little drop of water from them, they will flourish. And they live in dunes. So when you walk across a dune and realize that there's a crust that breaks up, that's because of all the microbes that are binding the sand grains together that are forming that crust. And on desert rocks, there's a phenomenon called desert varnish, which is a kind of coating of iron oxides and other metallic oxides. And the desert varnishes are a habitat for a specialized group of bacteria that are thought to be part of the process for catalyzing that reaction. And the desert varnishes will form over many years. So if you go to the world's deserts, for example, I think that one was in Utah, you can see where the desert varnish has been used as a medium for creating artworks by the indigenous population there in times past. And that's all because of the work that the bacteria have done over the centuries. And it also highlights what those organisms have done. And desert varnishes have occurred through deserts throughout geological history. So, for example, on the south coast of England in desert, there are some sea cliffs near Budleigh Salterton, which were formed in the Triassic, which was a period of desertification. And there are layers in those cliffs where you can see lines of desert varnish, and you can think about the lost habitats the lost desert habitats with their bacteria, which once occurred in the British Isles. Now, desert bacteria some of them can be extraordinarily resilient. So, for example, there's a group of desert bacteria in the genus Deinococcus, and they're very resistant to ionizing radiation. They seem to have extremely competent DNA repair mechanisms, and they have multiple copies of their genomes to allow for the fact that they can be damaged in the extreme environment they're in. And no doubt these organisms get transported around whenever the deserts move around. So you'll probably be aware that sometimes when the climatic conditions, when the weather's in in a particular sort of stream up from the Sahara. You can get Saharan dust blown into England and into northern Europe. And there is some literature of looking at Saharan dust and looking at places like mountains in the Alps to show how actually that dust is a rich source of transportation of desert bacteria to different parts of the world. Now, I suppose that the point I'm making here is that bacteria move around. So if there's a habitat that's suitable for bacteria, they are able to travel. However specialized they are, they seem to be able to get from one place to another. There are bacteria that thrive in very cold conditions. And I did a project with Anna some years ago. It was one of the first ones we did, I think, where Anna had got in touch with an institute in the north of Finland, and we worked with, she was working with somebody called Minna Minister, and she sent us some of her strains of Arctic bacteria that live in very cold conditions, and we were able to grow them up in the lab, and that resulted in an ice sculpture depicting the bacterium. They can also live in unusually acidic conditions. There's on Anglesey, which is in North Wales, there's a massive area of ancient copper mines where the copper chemistry has caused the aftermath of the mining has caused the water bodies there to be at the same acidity as battery acid. And even the rocks themselves, apart from a few heather plants, there's not really much growing there. But if you look at under the microscope at the water of these ponds, it's teeming with acidophiles. Wonderful landscape to visit, actually. Okay, salt-loving bacteria, and most of which are actually archaea, to be strictly correct. One of them is really extraordinary. There's a thing called Quadratum wolsbei, or Wolsby's square-shaped. It was called Wolsby's square-shaped bacterium for a long time. It's actually an archaeon, but it's a prokaryote. And he was a microbiologist based in Bristol who was looking at this very solar Salton, a salt pool in Egypt, and found these weird square-shaped structures in it and realized that they were actually living things. And then since Wolsby's discovery, the same bug has been found at salt ponds in Spain and in Australia. And this illustrates what's known as the Pecking hypothesis, who was a Dutch microbiologist who said everything is everywhere but the environment selects. So basically, if you provide a place for bacteria that's going to suit them, they'll come. And that's a close-up of Wolsby's square-shaped bacterium, or archaeon. It's got little inclusions in it, little gas vacuoles, which I think gives it some sort of buoyancy when it's living in its salt ponds. And you can go to the place in Spain where it's been discovered and enjoy the habitat. You can go and watch the flamingos, and you can think about the square-shaped bacteria that are living in there. So, through periods of history, there have been very sort of dry times. The Permian was a pretty austere time. The Triassic was another time when big salt lakes have formed and dried out, and that's the origin of the big salt deposits that people mine for salt. So this is Bulby Salt Mine in Yorkshire, and managed to get permission to go there and collect some samples, and that's basically rock salt that's been hauled out from, I think, a sort of couple of miles deep down below the earth. And the odd thing is if you provide the right conditions for halophiles to grow, they will grow from those salt crystals. And this is an experiment that's been repeated many times from many different salt mines, that if you get rock samples from under the earth, you can grow halophiles from them. And there's some debate about whether those organisms have been there all the millions of years it's taken for them to be trapped in the salt, or whether these are just contaminants that have got in and colonized the salt mine. But it does open up the possibility that maybe they can survive deep down in under the earth as halophiles in those salty environments for hundreds of millions of years, which seems a bit strange. If that's true, then that gives us a lot of hope for life in the future in terms of what it might be able to tolerate. There are some beautiful salt lakes or soda lakes around the world that have salt-adapted or soda-adapted bacteria in them, and one of them is Lake Magadi. I worked in Kenya for a while as a medical microbiologist, but it was always wonderful to go down to Magadi and enjoy the purple salt lake. And this is due to a photosynthetic purple halobacterium that's got purple pigment. And these are the pigments that get into the shrimps that get into colour the flamingos. Hot springs have their own flora. The scientist who realized that there were things living in boiling water and basically set up the science of looking at thermophiles was an American called Thomas Brock. And you can go to his study site in Yellowstone, and you can see the bacterial mats there, which are widely advertised as tourist attractions. And they're extraordinarily beautiful places. And that's basically bacteria that you're looking at there. Similarly, you can go to Iceland, and there are hot springs there which have similar communities of bacteria, closely related to the ones in Yellowstone. And even the hot pipes that the Icelanders use for their heating supplies are colonized with a bacterium called Thermus scotoductus, which specializes in living in hot water pipes. And it's even been found in the hot water supply of the London hospital. So again, if you provide the right habitat, somehow they'll get there. And that's a picture from one of the hot springs in Iceland. So just to get back to who invented bacteria, of course, you will know that it was Antony van Leeuwenhoek. And he corresponded with Robert Hooke at the Royal Society. And from a British perspective, it's kind of interesting to think about the scientific community that was happening at that time in the late 1600s, that you had a scientist who'd realized that there were living things in samples that hadn't been seen before. And then he was corresponding with Robert Hooke, who was the microscopist who'd at about the same time had first described living cells, I think by looking at a piece of cork. And he repeated von Leeuwenhoek's experiments, and this was all written up in the Royal Society, and it was a big deal at the time. And then bizarrely, nobody took any notice for another 100 years or so. Until the great medical microbiologists came on the scene, like Pasteur and Robert Koch. And of course, they were interested in the entirely practical things about what was causing disease and trying to address questions about whether there was sort of spontaneous generation of organisms, and if not, what was causing disease, and pinning the blame on bacteria. And then there was a big gold rush looking to see what diseases were caused by bacteria and working out what they were and naming them all. But this was terribly bad news for bacteria, because everybody suddenly got the idea that they were out there trying to kill us all. But that's been good for me, because I've had a career as a medical microbiologist, and these are some of the medically important bacteria that I've worked with, and they come in different shapes and sizes. So traditionally, what we've been doing for the last 100 years, ever since Pasteur and Koch invented the techniques, is that we've been doing microscopy. We've been looking to see what shape and what stains are absorbed by the bacteria in order to try and get an idea of how to identify them. We've been using Gram Stain, who was a Danish pathologist who developed a staining method for bacteria. And we've been using growing techniques which aren't terribly different from what Robert Koch did, except that we've since discovered antibiotics and are able to put up sensitivity plates to test antibiotic susceptibility. And that's basically what we've been doing for the last 100 years. And then I had this amazing good fortune about 10 years ago to get involved in a research consortium called Modernizing Medical Microbiology, because it was at about that time that sequencing technologies to be able to look minutely at the actual coding of DNA started to become affordable, and I was able to join a multi-center group to start looking at sequencing as a way to develop new ways of modern ways of looking at medical microbiology. And what became the mantra after we'd sat down for a bit and thought about this, what became the mantra very quickly was, if you've got the whole genome sequence, you can look at the resistance genes, and so without even having to do a formal test, you can predict what it's going to be sensitive to. And you can also look at how the bugs are related to each other. So if you're interested in an outbreak in a hospital or a food poisoning outbreak or whatever, or trying to understand where people are getting their TB infection, their tuberculosis infections, all you have to do is one test, which is to sequence the bug, and you have a single method that allows you to do everything that you want. So that became the mantra of single test to do everything. And so I had the fun of being involved in that research to look at how we could test the utility of this method to look at transmission and outbreak detection and resistance with one method. And whereas the old gram stains and everything have a certain poetry about them, the modern sequencing lab is really a bit boring because you've just got some machines to extract DNA from organisms and then run it through a machine, and then you get a lot of data that has to go off to a mathematician to work out what it means. But it does have a fun side to it. So, I mean, I was based in, spent a lot of my time based in Brighton, which looked like it would be a great place to try and understand gonorrhea better. So gonorrhea is a sexually transmitted infection, and it's caused by a bacterium. And there is public health interest in it because there are increasing rates of antibiotic resistance, so it's becoming harder to treat. And it tends to be most common, most prevalent in cities, and I happen to be based in one on the south coast that had plenty of cases for us to look at. So we were able to set up a method and look at gonorrhea, and I really look at it in detail for the first time, I suppose, in that respect, because until that time, all we could do was test a patient, find out if they've got it, treat them, find out what it was susceptible to, and that was about it. But we could actually start looking at the molecular epidemiology of the bug, and so when we started looking at the genetic signatures of the bug, what we find is that quite a lot of patients who've got gonorrhea are part of two different outbreaks, because they might have two different body sites. They might have two completely different strains of gonorrhea, and that's already starting to tell you something about the sort of the networks that are involved in transmission of this infection. And then if you look at the profile, for example, of cases in Brighton and how the bugs are related to each other. So if you imagine that each of those dots is a case, and then that you're looking at the accumulated cases over time, and as you go up the graph, that's each space on the way up is a new infection arriving. You can see that some infections arrive in the city, and they get passed on to other people and they become established for a while. But all the time you've got new introductions going on. So it gives you a sort of picture of how the disease is arriving and then how it's being transmitted within the population. And we didn't just look at Brighton isolates. We were able to share data with an American research group and look at the relatedness of our strains with strains from America. And some of them were so close that we could reasonably reliably say that they were part of the same infection network, and that the transmission had occurred within, well, certainly within the last 6 months to a year. In other words, this is it's a global transmission network that we're looking at. And what's highlighted there are strains of the gonorrhea bacterium that have a particular gene that encodes penicillin resistance or cephalosporin resistance, which is of interest in terms of tracking antibiotic-resistant gonorrhea. And then in fact, looking at things on the big tree, you don't need to look at that in detail, but that's basically looking at the whole collection. And you can see it's a dog's dinner of lots of different unrelated strains, but there are little clusters that cluster together. So some of them are Brighton ones, and some of them we got from friends in London, some are American. And then we got some strains from parts of the country where there isn't very much gonorrhea to try and understand whether there was something different going on in those parts of the country. So the ones on the left are from Northumberland, which is in northeast England. And basically, they've just got a single outbreak. They've got one strain. They're cut off from the rest of the world. So as far as gonorrhea is concerned, Brighton is closer to America than it is to Newcastle. But then when you look at Wales, not many cases from the collection that we got from Wales, but they've got lots of little mini outbreaks going on. So, Wales is obviously connected with quite a few other places. So it tells you what the humans are doing, basically, is what that's telling you. And that's just some references. And then tuberculosis is a very important disease globally. It causes TB. It's one of the problems with tuberculosis is a very slow-growing bacterium. So in order to find out what the optimal antibiotic treatment for the patient is, you have to grow the bug for a period of about 6 weeks just to get it growing, and then you've got to set up resistance plates for another 6 weeks to get the result. And by then, of course, if the patient's got a resistant tuberculosis infection, you've wasted maybe 12 weeks or more. So wouldn't it be great if you could just sequence the bug as it's just starting to grow, haul the genes out and say, well, it's sensitive or resistant to such and such. And the other thing about tuberculosis is that by necessity, the management of tuberculosis in terms of finding cases and looking at contacts and trying to close down the infection by controlling its transmission is done by local groups of TB nurses. And wouldn't it be great if you could get all of your strains from the whole country and see how they're all joined up and see whether people are moving around the country with the infection? So that's what sequencing lent itself to. And so most of this work was done by a team in Oxford and Birmingham, actually, but Brighton was one of the study sites where we were able to gather cases. And very quickly we started getting these fascinating results showing that a case in Brighton was very clearly part of the same outbreak as a patient in Birmingham, as a patient in Leeds. And suddenly it all starts to join up, and you can start thinking, what's going on here? And the TB nurses can go out and get information and find out what it is that those people have in common. And very often it's because they live, I think the term is that they live Celtic lifestyles. They're all part of some group of alcoholics or whatever that go to the same hostel. So there's some sort of reason that pins them together. They've got a TB risk, which is social, as well as having TB. And for the first time, you can start to put that in concrete scientific terms and think, well, what can we do to improve that? So another bug we looked at was Staphylococcus aureus, which on microscopy looks like a bunch of grapes. And you've probably heard of a thing called MRSA, which is a resistant form of Staphylococcus aureus. And the Dutch have put a lot of effort into monitoring and dealing with MRSA. And the Netherlands is one of the countries that's produced a lot of important literature on this organism. But we've certainly had our problems with it. The term Staphylococcus was first used by a Scottish surgeon who was looking at causes of post-surgical infection, Alexander Ogston. And we did a study in Brighton looking at our intensive care unit. And of course, one of the questions when you're doing infection control is thinking, well, it's an organism that colonizes humans. It can cause infection, but usually it's just colonizing us. And it's very easy to think, well, is it the nurses and doctors that are giving the bug to the patient? So we thought, that's fine. We'll spend 14 months swabbing the doctors and nurses every month, so we know exactly what they've got, and then we'll collect all the Staphylococcus aureus that is in the patients, and we'll see how they're joined up by doing sequencing. And I might add to this that this was under conditions where we were doing actually pretty good infection control. But what you see, looking at the unit, if you look at the line on the left, the steep line is, each of those dots is a new strain coming in. And when you've got lines going across, that's because we're seeing the same strain on the unit over the course of those months. And actually most of those lines is because it's a healthcare worker who's just colonized with the same bug. What we can see is Staph aureus strains. They're not moving around the unit very much. We're getting new ones coming in all the time. So every time a new patient's come in, they're coming in with their new Staph aureus. So in effect, that gave us some confidence to say, well, although we get occasional infections in patients, they're coming in with their own Staph aureus, and we can back that up without scientific evidence from the sequencing. So that's what that was about, and that gave us a lot of confidence that what we were doing was right. Anyway, enough of that. What Anna had asked me to do was to talk about the end of the world and a future without us. So eschatology, I think, is the term that theologians give to the end of the world idea. And it's always topical, and it must always be topical, because I think it's self-evident that life ends, but it goes on. And it's fragile, but it goes on. And it's self-evident that at all kinds of levels there are cycles of death and rebirth. And James Joyce said that's the point of eschatology. What can be coded can be decoded. I don't know what that means, because that's not a real word. But this is from his famous book, Finnegan's Wake, which is itself is a book of death and rebirth, because it's a cyclical book. When you get to the end of the last page, the sentence runs on to the beginning of the first page. And even the title itself is ambiguous, because if you just say Finnegan's Wake, it sounds like a wake is a party at the time of a funeral. So it sounds like somebody called Finnegan has died. But there's no apostrophe, so it actually probably means that the Finnegans are waking up. And you can compare and contrast cosmic cycles. So the system which is closest to what modern astronomers might conceive as being reasonable is Vedic cosmology, which is a time of where time is infinite, and there are long 4 billion year cycles which keep on repeating themselves. Or if you're a Northern European, in pagan times, you might have adhered more to the idea of a final disaster where a couple of people survive by hiding in a wood and they go on and repopulate the earth again. So, every culture seems to have its idea of disaster and cycles. And John Carpenter's famous film, Dark Star, has a kind of cycle in that the Phoenix asteroids circle the universe every 12.3 trillion years. And bacteria have cycles. Escherichia coli replicates every 20 minutes. So they're conscious of this too. So the prevalent scheme in Western Christian thought for the last couple of thousand years has been is the ideas encapsulated in the Book of Revelation as recorded by John of Patmos, and there he is looking remarkably consistent through art history, looking very relaxed about it, because this I guess is the way of things. Or put it another way, the avant jazz musician Sun Ra said it's already happened. It's already after the end of the world. One of the lyrics from this album is, "It's after the end of the world, don't you know that yet?" So, just to finish off, when the Earth becomes uninhabitable for humans, and when it even starts to become uninhabitable for bacteria, what chance have they got to go somewhere else? So there is quite a lot of interest in survival of bacteria in space for various reasons, but from the point of view of this talk, let's just mention that there is an account of exposing bacteria that are embedded in salt crystals on the outside of a spacecraft, and they've survived a space flight. And then we know that rocks can travel between planets, so there is some optimism that maybe the bacteria will make it. So to summarize, life in the future, this is just going back to the same slide again. We know that they're diverse. We know that they can tolerate terrible extremes. We know that they can flourish in all sorts of strange circumstances, that they can survive dormant as spores or whatever. So in the future, I think they'll have many opportunities. And just some brief acknowledgements from all the amazing colleagues I've had. That's just some of them. And I'm just a little bit that's hitched a ride on this story.
Thank you. That was amazing, I think, anyway, because I spent the last 15 years like going, wow,
Well,
to literally everything that John ever tells me. I would like to take some questions then for John. Has anyone got any questions? Is everyone really shy at the moment? Anyone? Because I've got one. I've got a few, actually, but I can start with one whilst they're warming up. You talked about the gonorrhea transmission networks, what that revealed, and I know the National Collection of Type Cultures has now acquired for me to make some art with the super gonorrhea strain that's been in the news recently. Yeah. But one of the important things with whole genome sequencing is that it reveals these things about where we are, where we're going, where we travel to, and those sort of privacy issues, which we've been talking about over the last day or so, about this thing about, do we own our own data, do we own our own privacy? And there are privacy issues in whole genome sequencing of bacteria, just the bacteria that live on us, because it's so complex. Could you say something more about that?
Yeah, so if you sequence a bacterium and it provides information that says something about you, then from an ethical point of view or from a legal point of view, there needs to be some way of making it impossible, or at least very difficult, to link that information with you. So, for example, with gonorrhea, with the studies we did, it was fairly straightforward in that everything is anonymized. And we did get permission from patients if we were getting information, behaviour information for them, which was one of the sub-studies we did. But what you would normally do would be to ensure that you know about the bug, you know some very minimal bits of information about it, like it was cultured at a particular date in a particular city, but that's as far as in terms of making the sequencing information available more widely. That's all anybody else would get. And just to say a little bit more about that, a huge amount of thought's gone into this. One of the problems when you sequence bugs from a clinical sample, that there is just a risk that human DNA might get into that sample, and then if you upload the reads into a publicly available database, it might be possible to work out some information about the person that the bug came from. So there are fairly carefully thought through protocols for removing DNA, both at the extraction step and also at the data, at the pipeline, as we call it, at the stage where the sequencing reads are being analysed. If there's any evidence that some of those reads look like human DNA, they're filtered out and removed.
But some humans have more susceptibility to certain bacteria, and obviously by having access to the DNA data, that might also, the human DNA data, might reveal things about who has susceptibility and who doesn't as well in the future.
So indeed, so there is a human reason to want to have more information than what I've just described, but then you have to go through some ethical hoops to allow you to do that. That might might involve getting informed consent for whatever in studies or whatever.
We had informed consent for my work with gut bacteria from the Oxford patients, who were apparently all very keen on having an artist using the data from their gut bacteria and actually using their gut bacteria. So it was a good conversation starter, apparently, with the patients. Is there any questions? Yeah, Chris.
Hello. Sorry to bring it back to the vulgar, really, but what is your favourite bacteria and why?
Oh, I don't think anyone has ever... Has anyone... Well, maybe you've asked me, but not for a while. What's my favourite bacterium? Just off the top of my head, I'd probably say Staph aureus at the moment, actually, because it's an easy thing to work with. A lot of bacteria try and commit suicide when you try and grow them in a lab.
Gonorrhea.
And,
that's bad. That's hard to work with.
I spend a lot of time working with Streptococcus pneumoniae, for example, and they're quite fussy. But Staph aureus, you can go away for the weekend and they're still quite happy. And then, it's a fun thing to work with.
Thank you. Any other questions? Yeah. Oh, well, loads now. So should we start here? I will move around.
I was just wondering, because you mentioned like the 3 branches of life and also the possibility that bacteria survive quite a lot. So just like in the scenario that everything dies except bacteria, whether there's like the possibility that from bacteria something other evolves than just bacteria, or they are once evolved in their path, there's no going back from being a bacteria.
Well, I think if you accept the findings from evolutionary biology and phylogenetics, we're derived from bacteria, basically. So no reason why it shouldn't happen again, except that conditions are going to be different. And I suppose from a Vedic cosmology point of view, you can imagine that maybe there have been an infinite number of experiments and it's been different each time.
And the other questions? Yeah, Paul?
On one of your slides, you had the term, I think, lithopanspermia, and you didn't describe it, and it's new to me. Could you explain what it is?
Okay. So it's just the idea that I think what's gaining favour with some space biologists or whatever is that the best chance for microbes to travel between planets is if they're embedded in big chunks of rock, because it seems like they don't last very long when they're exposed to cosmic radiation. It looks like maybe there is a chance if they're in the middle of a big rock. So that just basically means seeding by rock.
Other questions? We have some more. Yeah.
Yeah, sorry, I forgot your name. What's your name?
Me, Anna.
Anna. Okay, earlier you talked about that you use bacteria in your artwork, like real life bacteria.
Yep, pathogenic organisms as well, from tuberculosis and Yersinia pestis plague to the lovely Staph aureus, which we collaborated on, all sorts of things.
Yeah.
Clostridium difficile biofilm sculptures, which are currently on show in an exhibition curated by Angelique at the back at Moo Gallery in Eindhoven, for instance.
I was wondering, because you use bacteria that are related to diseases, whether that isn't dangerous or anything, whether you can just use them and it's okay.
Well, it's not dangerous if you use the proper methods for doing it. So I work with senior microbiologist John, and I work with Public Health England, the National Collection of Type Cultures, and actually over many years, because, I mean, when I first started to do it, I was like, can I do this, can I do this? And we had to work out, and John's very open-minded, so he really thought about this, because one traditional way of, like with science things, like, no, you can't do that, no. They would just automatically say a default no, but you didn't say a default no. You thought about, well, what can we do? And if we want to show things like, if we want to make art with Staphylococcus aureus, well, and this was a project with James Price, who was a co-author, the lead author on that paper that you showed about the Staph aureus transmission in intensive care units. I said, well, he said, well, we're growing it on this pigmented agar that makes the bacteria grow blue. And I was like, well, what do you think if we sterilise that in a high pressure sterilisation thing? And he was like, oh, nobody's ever tried to do that. So we tried to do it, and it stayed blue, and it looks fabulous. So we did that. We made these artworks that used the patterns of the antibiotics to make quilts and things like that. But we used the methods, and now I wanted to make art with tuberculosis, really the biggest challenge to microbiology ever, really. It's one of these things. It's such a clever organism that gets around kind of every method that we ever kind of try to beat it. We think we've got a handle on it, and then it'll get around it. I wanted to make work with that, and I asked you about it, and you said. You said that, you know, there was some rule at first, wasn't there? It was actually under a bioterrorist ruling at first. At first, though,
right. So
I think.
The TB story, the tuberculosis story, is a good example of how we turned what looked like a problem into something that was actually good public engagement. And so tuberculosis is labelled a Category 3 organism, which means that it has to be handled in a particular kind of secure lab, and when you're handling it, you have to work with it under an extraction hood, and the bugs aren't allowed to come out of the lab unless they're very definitely dead, and even when they come out dead, they have to be sealed and taken away and disposed of in a particular way. And Anna said, I want some of these to put on my artwork. So the normal answer would be no, except that when we were setting up the sequencing study, one of our problems was that we had to think about how we were going to get DNA from the Category 3 lab into the sequencing lab so we could do the sequencing. So that was fine. We could do the DNA extraction method in the Category 3 lab, but then we had to show that it was dead. So we spent quite a long time doing kill experiments that lasted many months to satisfy the external safety inspectors, shall we say, that what was coming out was dead. And once we'd gone to all of those lengths, it occurred to me we'd done everything responsible to be comfortable that that could be used in an artwork, because it was good enough to take into an unsecure area and work with on the bench. So it was good enough for an artwork, because we,
And so,
as far as we knew, it was dead. And then that became part of the story with Anna's artwork as to what she'd actually done, was that we've had to develop new protocols, and this is what we've had to do the sequencing.
And then the National Collection of Type Cultures at PHE had to do a similar thing for Yersinia pestis, which is plague bacterium, and they use killed plague and then extract the DNA from that. And I was able to go and, because I ended up extracting the DNA from the tuberculosis and going through that experiment hands-on in the lab. And then I went to the National Collection of Type Cultures at Colindale Public Health England, and I extracted the DNA from Yersinia pestis in the lab with them. And that piece is now on show with Angelique at Moo Gallery in Eindhoven, so you can go and see the artwork that I made that has two of those. In fact, it's got Streptococcus and it's a Streptococcus pyogenes, which causes scarlet fever, and Corynebacterium diphtheriae that causes diphtheria around the neck as well. So it's this kind of 18th century nightgown. Actually, no, that's in China, isn't it? Yeah, she's curated lots of shows, and I get confused where they all are. But no, that one's in China. But there's other ones. The Clostridium difficile one, which was with Leeds University. And that's another thing, because Clostridium difficile has these spores, which are really hard to kill. So we had to do an even longer autoclaving process. But we managed to actually create three-dimensional sculptures from the biofilms of the Clostridium difficile bacteria, which is a superbug. And that's on show at Moo. So it's working with the methods that the scientists are using and understanding them. And also my questions, I think, it was like when I first asked to use TB, that wasn't quite happening, that stuff. And I'm kind of constantly asking. And so I was like, I want to get rid of an antibiotic resistance gene, because as part of the sequencing process, and understanding that, when I started working in Modernizing medical microbiology, I found that I had a Staph aureus that lives on me, lives in my nose. And so we extracted the DNA from that, and that went into the Oxford database. But also I've been working with that ever since. And I decided I wanted to kind of understand how to remove an antibiotic resistance gene from it. I still haven't managed to do that, but I did manage to remove an antibiotic resistance gene from an E. coli bacterium. So that's my first step on the way. But it's to keep trying to find people that can help me with it, because Staph aureus isn't very much used in CRISPR genetic engineering yet, because there's an issue. You either have labs that work with synthetic biology, or you have labs that work with infectious diseases, and the two don't come together very often, and if they do, people get a bit scared sometimes. But that's what I need for this artwork. But I'm sure I'll find it one day. And with the very finest health and safety restrictions on it. Other questions, please? John?
It's not really to do with the end of the world, but it is to do with bacteria. You were talking about a place, Santa Pola, and the lake there, the bacteria there. South of there, but further south of there, not too far, Torrevieja, there's a lake that's very pink, intensely pink, and there's flamencos there. I'll get confused.
There might be some flamencos.
There might be. There are both, I think. And I was wondering if I think, did you call them
halophiles?
Halophiles.
Salt lovers.
Right. Okay. Do you think that it's possible, or is this a known fact, that the flamingos transport
do they transport them?
That bacteria in their, say, their feathers?
I hadn't really thought of that, but yes, almost certainly, I would imagine.
Okay.
Yeah.
Simple yes or no. Great. Great.
You might be able to get a paper on that. If you start writing it up now.
Yes or no.
And Angelique had a question.
So it's one of the things I'm always wondering about. You said, like, we don't know all bacteria yet, and there's like trillions of them. One of the worrying points is biodiversity. Like, at what size does that matter? So, like, bacteria, really small, tiny, and they seem to be everywhere. And the bigger the organism grows, the harder it gets to survive. So what's the ideal size?
Ooh, that's not my area of expertise.
No?
I wouldn't even like to guess, but probably bacterium size.
Yeah.
Yeah.
Okay. That's what I wanted to hear, Finky.
Other questions? One, I suppose we're sort of winding down. We've got about 5 minutes left. So any other questions? Yeah, Arion?
Continuing on this idea of the lithotranspermia, I think it is. I'm thinking of how we found other continents, and often, I mean, brought our bacteria to these other continents and disrupted the habitat there and the life that was there. Do you see lithotransperia as an actual thing that you need to worry about? And I mean, yesterday it was presented as continuing our life on other planets. It could also be disrupting possible life on other planets with our bacteria. I'm very curious to hear your opinion on that.
All right. So I haven't developed a personal view on this, but some of the literature looking at viability of organisms in space has been done by NASA or funded by NASA because of concerns that we could transport bacteria to sterile planets and mess them up, or we could at the very least hinder opportunities to understand whether, say, Mars really does have...
Because NASA already now is taking care that there are no bacteria on any rockets or any spacecrafts going.
Yeah. So exactly. So that's around being able to test how confident we are that we're not transporting them to other planets. That's part of the reason behind that work. And so obviously the sort of ethical thinking that goes on in the space program has considered that and considers it to be at least a scientifically bad thing, because you don't want to mess up your search for life by taking it there and then confusing your results. But
that's another point.
There is maybe a moral issue about why should we put Earth life on another planet if it's already got its own things there. Although having said that, if it is possible to do that, then it's probably already happened.
One last question, or are we done with the questions? Shall I round up? Yeah, I'm going to round up. Actually, leading on from what Arion said, that idea of taking bacteria from one country to another, I had a very interesting conversation with Tim Walker at Oxford about this. And he said, from the sequencing data that they've looked at and has been looked at, there's a thing where we think that places like India, and where I'm shortly about to go, Uzbekistan and places, but mainly India, have these very high rates of TB. And so when people want to migrate to the UK or something, there's this whole thing about they're bringing TB to the UK, they're bringing TB to Europe. But actually the sequencing data shows that these strains that they have originated in England when colonialism started. So they've looked back at old samples, and the samples from sort of the colonial period in the UK and are actually related to the samples that they're seeing now in India. So if people do bring TB across, they're simply bringing it home. Which I thought was a very interesting perspective on this whole thing of colonialism and immigration and the other that we've been talking about.
Yeah, so the TB story, as far as I've looked at it, I mean, TB is an interesting organism in that it is well adapted to humans and it appears to have been with us for a long time. to the extent that the consensus is that we probably walked out of Africa with it. And that at which stage, when human populations were very sparse, it was probably quite a low-grade pathogen, because the interplay between a potential pathogen and its host doesn't favour lethal pathogens, because they wipe out the host in small communities. And it looks as though it has urbanisation started to happen in maybe the early civilizations. That's when the more virulent strains developed. And you can imagine a sort of a global map and lots of arrows with all the different TB lineages that have moved back and forth. And indeed, it does look as though strains have come out of Africa and then gone back in again as Europeans have gone there, and some of them, those are it's possibly because those are nastier ones that have developed in European cities and have been in a situation where it's the population biology favours a more lethal, a more virulent bug, and then it gets transported somewhere else. I don't think I've said that very well, but.
I was going to say, so on the first evening of this programme, I introduced a film called Genesis 2.0 that Arion programmed, which was about trying to bring back the woolly mammoth through synthetic biology and sequencing technology. And I'm reminded now that the earliest samples of tuberculosis that have been identified were in Pliocene-era mammoth bones. So we had a conversation earlier about who's to blame for this end of the world thing, and I think we can really, if anything, put the blame at the door of the mammoths. I'm going to finish coming full circle and saying it's all the mammoths' fault. Yeah.
They deserved it.
Yeah, you might want to think more about this and come back with some different debates, but I'm going to end it there, blaming the mammoths. And I want to thank everyone for coming, and I really want to thank John for his wonderful talk, all the panelists, and Paul Brown for his wonderful keynote last night. It's been amazing. It's been really interesting. I hope you all feel the same, and thank you all for coming. So thank you. And thank you to Arion for IMPAKT.